Hello everyone, welcome back to my YouTube channel. I'm Dr. Arpita De. Today we will learn a very important topic from developmental biology. We will study about vulva development in C. elegans. C. elegans is an organism chosen for the study of developmental biology because of its short life cycle and easy availability. So let's come to the topic. C. elegans individuals are hermaphrodites. It means what? You can study both the sexes in this organism in one single organism. They are male in their early developmental phase and the gonad produces sperm which is stored for later use. Ovaries develop as the nematode grow older. The eggs roll through the region of sperm storage, are fertilized inside the nematode, and then pass out of the body through the vulva. So, the vulva is an organ of primary importance. Here we will explore about how it is getting developed in the body of nematode. Here you can see a diagrammatic representation of the C. elegans body. The anchor cell here is governing these vulval precursor cells and will ultimately give rise to the complete vulva. Adult vulva comprises of 22 cells which are progeny of three ancestral cells originally located in a row on the undersurface of the developing nematode. Here you can see this one is the basement membrane and below here is the cuticle. So the cells are attached to the basement membrane and are being governed by the anchor cell. Before we go to the developmental process, let's first learn about how is this anchor cell developing. The cells of the equivalence group receives the extracellular growth factor like Lin3 protein signal. Before this, the anchor cell is formed which is mediated by Lin12 gene which is a homologue of Notch gene in Drosophila. In wild type C. elegans, two adjacent cells are there. One is Z1 triple P and the other is Z4 triple A. Both of these cells have the potential to become the anchor cell. They interact in a manner that causes one of these two cells to become the anchor cell and the other to become the precursor of uterine tissue during the second larval stage. Lin12 gene only functions in the cell that becomes the precursor of the uterine tissue. The presumptive anchor cell doesn't need it. So the precursor for the anchor cell will not need it. It doesn't need functional Lin12 gene. Originally, these two cells synthesize both the signal for uterine differentiation, that is LAG2 protein, which is homologous to delta in Drosophila, and the receptor for this molecule, that is Lin12 protein. During a particular time in larval development, the cell that by chance secrete more LAG2 causes its neighbor to cease the production of differentiation signal, that was LAG2, and increase production of Lin12 proteins. Thus the cell secreting LAG2 becomes gonadal anchor cell. The cell that receives the signal through its Lin12 protein becomes the ventral uterine precursor cell. I would request you to pause the video and take down the diagrams and take your notes as and when required. Look at this. Here you can see the anchor cell, and the proximity of these vulval precursor cell to the anchor cell will decide which fate will be assumed by which cell. So here you can see P6P is at closest proximity to the anchor cell. That means what? The lin3 protein that will be secreted by this anchor cell, the signal in the form of lin3 that will be given by the anchor cell will be given at a highest concentration to P6P. P6P by virtue of its close proximity to anchor cell will receive maximum concentration of lin3 and will adopt central vulval fate or first degree cell fate and will form central vulval cell. P5P and P7P are a little far away from the anchor cell and will receive the signal obviously, but it will be lesser in concentration. Therefore, they will adopt secondary cell fate and will form lateral vulval cells. So, this lin3 is the protein and it needs a receptor in these vulval precursor cell. What is the receptor? The receptor for lin3 is let23. Okay. Now let's talk about another signal. Anchor cell is signaling P6P through lin3. Now lin3 has got an inhibitory effect on the adjacent cells. This inhibition is mediated by what? This lateral inhibition is mediated through lin12. The lin12 protein is activated by the primary vulval lineage to stop the lateral vulval cells from forming central vulval cells. This inhibitory action is very important because if this inhibitory action is not there, what will happen? All the cells will adopt primary cell fate. This is not required. In case if lin3 is not there, if lin3 secretion is not there, what can happen? There will be no signaling cascade, right? All the VPCs or all the vulval precursor cells will adopt non-vulval fate, and what will they form? They will form hypodermis. So you can understand how important lin3 is. Okay? If lin3 expression is blocked, what will happen? The cells will form hypodermis. If by chance let3 receptors are deleted, what might happen? The same result will be seen. In that case, what will happen? Lin3 is there, but who will receive that? The receptor is not there. As a result, what will happen? All the cells will become hypodermis and no vulval cells are developed. Now let's see. Receptor for lin3 is let23 receptor tyrosine kinase. The signal is then transferred to the nucleus through the RTK-RAS pathway. Lin31 protein is the target of the kinase cascade. Phosphorylation of this protein in the nucleus causes it to lose its inhibitory protein partner and allow it to function as a transcription factor promoting vulval cell fates. So, if any of these three signals, be it lin3, let23, lin31 are blocked, no vulva will develop. In if P6P, P5P, and P7P are destroyed, what will happen? The nematode is still left with P3P, P4P, and P8P. The rest, three vulval precursor cells, will form the vulva because lead 23 is present in all these vulval precursor cells. Okay, so they are all capable of forming the vulva. Uh, if P6P, P5P, and P7P are absent, what will happen? The remaining three will form the vulval. The remaining three will form the entire organ, that is, the entire or complete vulva will be formed. I hope you have, so this is how we complete our lesson today. I hope you have learned something new. If you find it difficult or if you have any problem anywhere, please feel free to get back to me in the comment section and I will try my best to answer your doubts. From time to time, please pause the video and take down the informations. For books, you can refer to Developmental Biology by Gilbert. That is one of the finest books for developmental biology. Other than this, if you have any confusion, I'm again and again telling, please feel free to tell me. Okay, thank you so much for your kind attention and hope to see you soon. Bye bye.